I'm Steve Gendel, and I'm with the FDA. Hello, Rick. But my question is not for you. It's for Hillary. I'm curious to know, when you work on these methods, the methods themselves can be really wonderful, but it's really important to understand that whatever you're measuring is representative of the food out in the real world, which depends upon the way it's sampled and handled. Do you guys work with your customers on sampling plans and developing methods to make sure that you're looking at representative material in the lab?
Yes, we do that. Can you hear me? No. Oh, this one over here. Yeah, I just turned this one on. Yes. Any better? And actually, for instance, for nutrition labeling purposes, the FDA has guidelines on how you select from different lots in order to sample. So we do advise clients on how to go about sampling correctly in order to get a representative portion that we will end up with to test. So.
Mine here. My question is for Kathleen. As you know, over the past years, there was a lot of discussion in the soybean and inclusion of endogenous allergens. Ultimately, the document came out, and those were not included. I was wondering if you could just comment a little bit on the thought process within OECD about why those weren't included.
Thank you. That was a topic that did come up for that was a topic that did come up for discussion during the development of the soybean document, whether or not endogenous allergens should be included in that table. I think it's table 21. And there was a lot of discussion, and there was not consensus among the group to include it in that table. And so the decision of the group was to include it, as we always do, all of the information that we find in the text of the document, but that would not be part of the suggested constituents recommended to be analyzed.
Okay, Jean-Michel Val from INRA and EFSA. I would like to make some comments regarding Rick's presentation. Is it better like that? Yes. Rick has presented a lot of information, very interesting information, sometimes very complicated, and I would like to make some comments as to try to answer some of the questions you raised. Very last one. You raised the issue of hypoallergenic GM crop. That's completely different of what we are discussing right now. That would be a health claim, and health claim would need some substantiation, much more complicated than the risk assessment. Then I think this is a part of the discussion. Then you have presented very nice results you have performed regarding the analysis of the allergen repertoire of the GM versus the non-GM, for example, soybean, because this is a requirement for the risk assessor. And you say, look at, we have several differences, but we have no risk. And you suggested, but maybe I misunderstood you, that because you have difference and no risk, it's not necessary and not useful. Not at all. You have a lot of difference, you have no risk, and for what you have presented that have already been risk assessed, you never had question nor rejection. Why? Because you said EFSA has changed its mind and now asked for individual sera testing. And what you showed is very evident, is that the difference between the GM and the non-GM are much lower than the difference between one allergic patient's serum and the other. So the difference you see using serum 1 are completely opposite using serum 2. That's an example where the difference you have evidence are not biologically relevant. And that's all for the risk assessor. If you have used a pool of sera, then maybe you have got one pattern with big difference between the GM and the non-GM, only with one trial, and then you would have been asked question to explain why there is such a difference. But as you did, since the difference between one allergic patient and the other are not constant and may vary, this is only variability, not only variability of the allergen content of the plant, but variability of the human IG response, which is also highly variable. Then there is no risk of allergy. And that was the conclusion of the risk assessor.
Sorry, Jean-Michel, could I address that point? I'm not saying there's no risk, I'm saying there's no increased risk. So if I said no risk, I'm sorry, I misspoke. I'm saying there's no increased risk.
No specific increased risk. Yes, you're perfectly right. And that's my last comment. You said also at the very beginning, what is the question? The question is the GM crop as safe as the non-GM comparator? For allergenicity, it's exactly the same. Is it as allergenic as the other one? That's a question which is asked to the risk assessor. And that's a question. And you suggest that doing this study might not be necessary or useful because anyway, you can protect allergic consumer by avoidance or by labeling. The problem is that are risk management issues, and that's a risk management responsibility and the responsibility of regulators, of policymakers. That's not the responsibility of the risk assessor. The risk assessor, like EFSA, has to comply with the regulation. And the regulations say the allergenicity of GM crop shall be assessed. And to assess, you have to make, yes, I finished. And you have to assess the composition of allergen between and compare between the GM and the non-GM. And you have presented a lot of methods that are perfectly relevant and acceptable. But all of these methods use human allergy patient sera, which are very rare and precious. And what EFSA has recommended or suggested, because it's not a strong recommendation, is that to do exactly the same work, if you can avoid the use of human sera by using, for example, quantification or semi-quantification of endogenous allergen, this would be acceptable.
You've raised many interesting points, and we should probably schedule an hour discussion. Can we do that? And I think one of the most important questions you said, and I'm not a true risk assessor versus risk mitigation or risk manager person. Maybe there are some other people, including yourself, who are better at that, and Steve Gendo and other people. I think as I read it, and I worked at Monsanto for 7 years and UNL for 8 years, and I work for major food companies on questions about does this new food product represent a risk of allergy, is that we kind of have some very technical differences in terminology between assessing risk and assessing hazard and then managing those things. They're very difficult, but unfortunately the way, as I see it, somebody not in the position that the FDA is and others, is that we kind of have one system that does both at once. Because if you can never get it on the market because of a minor difference, how do you ever manage it? The truth is that we manage risk of allergy on an individual basis. If you're allergic to peanuts, you have to avoid peanuts. We don't say that you have to avoid this peanut, we have to avoid that peanut. The reality of food, the real world of food, whether it's in the U.S. or in Ghana, is that we don't have people running around testing different varieties or different processed foods and saying which variety of a given crop is used. So yes, there's a risk to the individual. It's very individualistic. We use individual serum based on the recommendations or the request of EFSA, as I understand it, because for the different projects we've worked on, we used individual serum for immunoblots. That helps tell you variation in this person's sensitivity, which proteins are they binding IgE to, not necessarily allergic to, but binding IgE to versus this person, and you saw from the few examples that I showed that those can be very different. Do we know that one person won't drop dead from binding IgE to one protein, the other person is okay and they can eat a handful of peanuts? No, we don't know that. There's a lot of individual variation. None of the methods we use, whether they use serum and IgE binding, are perfect. If we use proteomics, and those are not perfect either, because they're, depending on how you do them, you either do solution, digest, and LCMSMS, or you Malbitov, or you use gel, stain gels and pick the spots. They do not pick up the 100% of the sequence of each of those proteins. They don't necessarily tell you if they're glycosylated, if they're not glycosylated. There are fragments on the gel that you don't find that may or may not be due to proteolysis in the plant. You can have risk that you can't pick up by some of the other analytical methods. There is no perfect method. So we use IgE binding because from the case of diagnostic test evaluation, test material evaluation, those are the same methods that are used, and you use pooled sera. And you say for a population of 5 to 10 or 15 subjects who have proven allergy to peanut, if you put their sera together in a balanced pool method, is there a twofold difference in this diagnostic test material on lot A compared to lot B? If there's less than twofold variance, then you release it and you say that's good for the clinician to use in skin prick test and diagnosis. Food composition is very different, and so you can't take all of that into consideration. So I could agree that, you know, in some cases we could use individual sera, we could use pooled sera, we can use a method. We've done more extensive testing in most other labs, I think, that have done this. We use reducing and non-reducing 1D blots. We've used individual sera for those. We used individual sera for 2D blots. And for 3 of the studies, we've used pooled sera for ELISA inhibition, and that is the method of choice for release of diagnostic reagents in Europe and in the US. And we've also now on another product used individual direct binding, and we used material grown at 3 different, very different geographies to try and look at that. And I think it shows you that there is variation, both the genotype and the location and growth conditions contribute to some variance that the subject's IgE will pick up. Again, we have to get down to the reality of if you're allergic to soybean, you should avoid soybean. If you're not allergic to soybean, we have absolutely no evidence that says if you're not allergic to soybean and you eat this variety of soybean versus that variety of soybean, you will suddenly become allergic to soybean. So I don't know the perfect answer to your question, but
okay,
what is it? I don't know.
Right, it's no time and the place to have a complete debate on allergenicity. What I would really try to explain and to make it clear, what you say is sensible. If you are allergic to soybean, you should avoid soybean. The regulation, and that's the law, say a GM soybean should be assessed for its allergenicity. Whatever you think it might be useful or necessary or not, it is the law. The applicant has to do that. Full point. Maybe it's stupid, but Duralex, Sedlex, as we said in French. Okay?
No, I can understand.
Then after you can use any of the methods you use. That's not the problem. Yeah. But this
so
has
here
to be done.
is what we need to come up with. Here is what we need to come up with. We need to come up with a definition of how much difference is too much if we're going to do those tests, and we have absolutely no consensus about that.
That's why the assessment is necessary. If we knew a threshold, it would be very easy.
Excuse me.
I mean, the only thing would be to
we really have run out of time on this topic, and we have another questioner behind. But I can see that this could be a topic for another discussion, if you don't mind.
Ray Schlitter, Bayer. I'm also chair of the Food Biotechnology Committee, and I'm hearing comments about the regulators asked for this or the law is this. The purpose of this workshop and the purpose of ILSI as a whole is to advise on the scientific situation, and if it doesn't agree with laws of countries or regulations, or what people may want to put into laws, that is not the issue here. The issue here is what is the science, and in all respect, what is the science of this area of composition and et cetera. I just wanted to remind people that that's what we are trying to discuss. And we do run up against what regulations have been done. We do run up against those things, and we try and put those on one side and say, what is the scientific situation? So I just wanted to get up here and say that, because I could see things are going off a little bit into some other discussions, which, in all respect, I know that it's an issue. Is that? Thank you. And that's coming. And this is the ILSI mandate, I would say. Thank you.
Okay, we would like to have some other questions from the group. Yeah.
I'm Kate Walker. I work for ILSI IFBiC. Hillary, you were talking about the validation of methods and so on, and yesterday Matthew was talking about the importance of sample preparation. And when I think about the whole range of, and you talked also about method development, how methods have changed over the years so that methods that were used several years ago might not be used today for the same analysis. So do we have any measure of how compositional analysis methods, how the results that we obtained from those methods, if you did a method from 5 years ago and a method from today and compare the results, would you expect to get the same answer, the same range of answers on the same samples, or is there creep in the results as the methods have changed, or sample techniques have changed, or sample preservation techniques have changed? How confident are we that the compositional analysis data we have are meaningful if the methods and sampling methods have changed?
Well, number one, I don't think they change. Well, in a short period of time, they typically don't change that drastically. Now, some of the methods, I believe, may reference all the way back to was it the lectin method, going into the 50s or something very, very old. Now, in many cases, some of the methods are, for instance, for a lot of amino acid methods, we use the same principle in the analysis, but we may have updated instrumentation, so we see a lot of consistency. Now, where you're changing from one complete category of method going to something instrumental, you may have a huge difference, in part not just because it's more so because we can address the issues that we talked about in the method validation, that it's more specific, you have better precision, all of those characteristics that you're looking for. So it very much depends. Some of the very old methods are very good and very consistent. We've been using them for years and years. Some of them, though, we don't believe to be too real accurate. There's a huge amount of variability. They're very old. And there is a need, I think, to do some collaborative work to determine exactly which is the best method.
And is there any measure of how much of the variability in the results comes from the crop and how much from the test?
Again, that's one of those things where I think what we have to look at is, because, yes, you can have huge variability, especially with the different crops, when it was grown, where it was grown, what country it was grown in. Now, what we would look at in terms of, for us, the method, again, becomes what controls we use consistently as we run over time. So when we see change in that, we know there's an issue to address. So two different tools to measure that.
Thanks.
Mm-hmm. Okay, the next person.
Is it on? Okay. Is it on? Okay. This question is for Hillary. I was just wondering, in your experience with looking at data over time, and maybe some of this is before your time with the company, but have you got a sense of from looking at historical values, maybe with the changes in the environment or just changes in soil conditions, like, I mean, is there some historical values that are no longer applicable? Like, do you just see a sense that sometimes they're just falling out of date and they're not comparable?
Again, it depends on the area you're looking at. Because we have so many GM soybeans and varieties that are out there, it used to be there was a time when I would look at a report for how much sense it makes before we send to a client. You knew that certain components like a protein was always in a certain range. Well, now there's a lot more variation because you have some of the GM varieties that are out there with the conventional. Most of them don't really change a lot. Again, it goes back to where they've been grown, at what point in time we've tested the product because of degradation, possibly.
I wasn't sure if there was sort of like a creep in nutrients, like something that was always a really good source of, let's say, vitamin K, like if the level was sort of gradually going down over time, or
no, and that would mostly be again degradation, and that would be with a particular sample that, you know, if you test it in a stability study, day 0 you're going to have your peak, and usually you keep testing it over a longer period of time, it will start to degrade, especially your water-soluble vitamins or vitamin C will degrade very quickly. But no, not that I'm aware of. And again, it would be very difficult to measure because there is so much inherent variability based on how long the sample has been sitting before we analyze it, where it was grown, and when it was grown.
Okay.
So.
If I could make a comment just as a complement to Hillary's talk, or Hillary's answer there. What we've seen in the Nutrient Data Bank over the years is that just in the last, I think 15 years is really, we're seeing great changes even in 15 years, where 15 to 20 years ago people were talking about total carotenes. Now they want individual carotenoids. We were talking about total folate. Now we want the individual vitamins of folic acid, folate. So there's a lot more specificity requested by the users of the data. It's what we're seeing, which impacts, I think, the laboratories. Certainly the government laboratories are impacted by that. I just wanted to throw that in. But, okay, Will, your next question.
Yeah, this is Will Ruby. I'd just like to comment on the last question. If you go back on the ILSI database, it's possible to search data for particular analyte by year, by geography, and by method. So there's a lot of information there that might be mined in order to address some of these questions about whether values for a particular analyte have changed over the years. The amount of information at an individual year can vary, however. I think the range, some of the database people can correct me, I think it maybe starts at 1995 and goes through 2004, 2005 now. There's much more data to be added and hopefully provide more information on how things have changed with time.
Yeah, and I've noted in addressing that, I've noticed when I've pulled some of the information out of the ILSI database, looking at some of the ranges for certain analytes, are huge amount of variation. Some are much more consistent, but there's a lot of variation just the nature of the analyte.
Okay, we have 1 more minute till close down here. So is there one question? Yeah. Okay, if it's brief.
Well, the question can be brief. I don't know about the answer.
The answer should be brief, too.
Yes, Steve Gendel again. So maybe this is a little off topic, but since people are getting ready for dinner, one of the things I was thinking about listening to you talking about variation in nutrients is when companies do their nutritional fats panels, is my understanding is what they do is take a recipe and throw it into a computer program that uses average nutrient levels for different kinds of foods to be calculating those panels. Do you think that there might be an issue with the accuracy of that information based on the variabilities that you're seeing?
Possibly. I know what programs you're referring to, and we actually do offer that as a potential service if somebody wants that, because it is certainly much cheaper than having to go through all the battery of tests. And most of those databases that have that information are based on laboratory analyses, like what you would with the USDA nutrient database. It is much more accurate, in my opinion, to actually go through with the analytical procedures to obtain that data. And I think one of the problems too is that you lose, since you're taking just ingredients and throwing them together and mixing them to come out with a final product for a nutrition facts panel that doesn't consider how ingredients react with each other, any cooking, the different variables that go from raw ingredient to what you eat. So.